Hi everybody, in this video we're going to cover signal reception and transduction. Really what we're talking about is how a cell knows what it wants to do, or what it should do. So, if you're a muscle cell, you can contract. If you're a nerve cell, you can fire. If you are a beta cell in the pancreas, you can release insulin. And any cell can grow and divide and or self-destruct. So how does a cell know which of these various activities to do at any given time? Now, what you're going to find as we go through this course is that a lot of this is determined by what kind of chemical signals that cell receives. Now, the ones we're going to cover today are shown in orange, and these are going to be peptide hormones. And these are some of the most common ones you've heard of, whether it's epinephrine, also known as adrenaline, or insulin. These are just really common signaling molecules in our bloodstream. Now, these peptide hormones are basically small proteins, maybe 30 to 100 amino acids in length, and they are polar. They will not be able to pass through a phospholipid bilayer, so the closest they can get to the cell interior is to bind to a protein that is in the cell membrane itself. Okay, so this is going to be the step called signal reception, and this receptor is going to be called a receptor tyrosine kinase. Now, there are lots of different types of receptors. This is probably the easiest one to understand, so that's what we're starting with. It's definitely not the most common, so word of warning on that. But this is the one we're going to start with. So, a receptor tyrosine kinase is a pretty sizable protein in the membrane, and it has three portions. One, it has this catcher's mitt that's specifically sort of shaped to bind to that hormone, and each one's going to have a different shape. There's the portion that goes through the membrane. That would be called the transmembrane region. And then there's this region on the inside that is called a kinase. Now, this is probably a protein type you've never heard of before, at least before this course. And it is probably one of the most important sets of proteins in life. A kinase is specifically an enzyme that can add phosphates to other proteins. So a protein kinase, again, takes a protein, target, say this green triangle here, and takes a phosphate from ATP and adds it to that target protein. That, of course, hopefully you realize is going to change that protein's shape a little bit and maybe change it from an inactive shape to an active shape, or perhaps the opposite. In this example, we're going to assume that we've just turned on the green triangle. Things to keep in mind. One, the receptor tyrosine kinase, the RTK, is an enzyme, so it doesn't just do this once. It can do this over and over and over again. So even though the RTK receptor has only received 1 or 2 hormone molecules, it might activate 6 or 10 or 200 of these green triangle proteins. Now, this is not the end of the pathway. It continues. What we're going to eventually do is try to result in some change in cellular activity, which might be in the nucleus, might be in the cytosol. But let's start with this green triangle. I'm going to assume, in a common manner, that this is a sort of common result, that this green triangle is another kinase. So it has the ability to remove a phosphate from ATP and add it to its target. Its target happens to be this whatever orange box. And let's imagine that this orange box is another kinase protein, and it can take phosphates off of ATP and add them to its targets. Again, the importance of this is that each step can do this more than once. So each kinase can phosphorylate, add a phosphate to, more than one target. So RTK activates lots of green triangle. Each green triangle activates lots of orange box. Each orange box activates lots of purple circle. Okay, why do we care? This is called amplification because we're going from maybe 1 or 2 hormone molecules up to maybe 100 or 300 or 1,000 final active proteins in the end, which might be these purple circles at the bottom. This is specifically called a kinase cascade because it's a series of kinases activating one another in sequence. And this is one version of what's called a signal transduction pathway. And if you go to the Greek, the Latin, transduction is basically to lead across, right? So this is going to lead the signal from the membrane where the hormone was bound all the way to the final target, which might be inside the nucleus or somewhere in the cytosol. Okay, so what's going to happen next? So we've gone from reception, where we catch the signal. We've now moved the signal and amplified the signal into the cytosol. Now this last target, wherever it might be the third, fourth, fifth target in the cascade, whatever the final target is, might have an impact inside the nucleus, or it might have an impact in the cytosol. And the most common version that we're going to talk about in terms of what the cell is going to do is turning on some genes. But it's also possible that the target protein in the end is just an enzyme that does something in the cytosol. That's the poorly drawn thing on the top of the diagram. Or maybe the protein triggers exocytosis or causes release of a hormone or causes muscle contraction. I mean, there are lots of possibilities here. But the one we're going to focus on, though, for this class is the fact that sometimes these final target proteins are known as transcription factors. And in the nucleus, they have the ability to regulate, turn on or turn off certain genes. So again, we've started with a signal in the bloodstream being received by a receptor at the membrane. Then we amplify a signal through the cytosol, making the signal more powerful and ending up in lots and lots of certain proteins being activated, in this case, the purple circles. And the end result might be turning on an enzyme or exocytosing another hormone. But for this class, we're going to focus on the fact that we might be able to turn on a transcription factor, which goes through the nuclear envelope into the nucleus and turns on or off multiple genes. That's where you get the biggest impact, where the cell starts doing an activity it wasn't doing before. Hopefully that gives you a good overview. We'll continue on and talk about how this impacts cell cycling and other pathways, including GPCRs, G protein coupled receptor pathways, in future videos.